This is Cindy Jacobs, and Jane Gross and I welcome you to the Business of Science. Stephanie Wisner will be discussing building strong scientific biotech companies for the next hour. Just before we turn it over to Stephanie, Jane and I would like to just kind of go through a few slides for women in bio. Stephanie, can you go to our first slide? Is just acknowledging the land, the traditional lands of the tribal nations that we are on. Next slide. And obviously, since a lot of us are in EWIB, we know that our mission, basically, we are committed to promoting careers, leadership, and entrepreneurship of all women in life sciences. And actually, my favorite slide is the next slide. It's kind of, I call it the circle of life. The circle of careers, basically helping from young women getting interested in careers in science, technology, engineering, and math, to helping young entrepreneur women in their, as this topic will go through as far as building companies, to C-suite engagement, to getting women into the executive C-suite or senior executive positions, and also then training for being a director on a board. Jane and I are actually trying to establish the Executive Women in Bio in the Seattle area, and both Jane and I have been through the executive training and boardroom ready and are now on boards thanks to that training. I'd like to turn over to Jane. You want to go through the remainder?
Yeah, so EWIB is Executive Women in Bio. It is a resource within each chapter. I think there is an EWIB portion in each chapter of WIB across the nation, or at least that's the goal. And that is to support and empower women leaders at the executive level. And it's really technically defined as a vice president, executive director, and above. Obviously, we're championing diversity and really trying to promote the presence of women in the C-suite. Okay, next slide. Some of the events are posted on our Seattle website, or you can find them on the national website. Some are open to everyone across the nation, and some are only local. Here are a few events that are going to be coming up. Connecticut EWIB is sponsoring a meeting in May called Building Talent Development Strategy and Empowering Action for Women. And then there are two events at the Bio Convention in San Diego that you can attend. One is a breakfast, which I've been to, which is great, and then the plenary session. Okay, next slide. And then obviously our vision for the future is to continue to support diversity. One of the objectives within boards today are to diversify, put women on boards and people of other diversities. Okay, next slide. And then here are some of our sponsors. A lot of them are local. For the Seattle chapter, Lyle, BMS, Amerisana, Zymeworks, Fred Hutch, Alpine, Amazon, Impel, and Umoja. So thank you to our supporters. And then tonight, this is our first event for 2022. We typically have 4 events per year. We hope to have a wine tasting event in person in June, and then often we have an event at Cindy's farm in Fall City, and then finally another event in the fall that may be more topical around business or business strategy or other topics of interest. And what we've heard from people is that they're interested in networking as well as seeing topics such as this. And I think this is, and I'm here to introduce our speaker for tonight, which is Stephanie Wisner. She's the co-founder and chief business officer of Centavax, and was listed as the 2022 Forbes 30 Under 30 in healthcare. And her background is really around promoting the information or the business for the scientific person within an organization. And so we're going to focus on how leaders are involved in building a better biotech, how they can strengthen their awareness of business and commercialization strategies to ensure a successful biotech. And Stephanie is going to give a small presentation, and then we're going to open it up for discussion. And so we hope that this is a interactive discussion and meeting, and probably maybe hold your questions until the end of Stephanie's presentation. I should note that Stephanie now has published her first book called Building Backwards to Biotech: The Power of Entrepreneurship to Drive Cutting Edge Science to Market. And it's available in Amazon. So welcome, Stephanie, and thank you for hosting this event today.
Thanks so much, Jane and Cindy and everyone for coming. I'm very excited to get to speak with you all. Jane gave a really great introduction, but just to give a little bit more color about how I ended up in biotech and how I got to where I am in writing books and stuff. I just want to tell you a little bit more about my story, because hopefully it's probably pretty relatable for all of you guys. So I did my bachelor's degree in chemistry and chemical biology at Cornell University. And I, like probably lots of people, thought I would go be a medical doctor afterwards, or go get my PhD, or both. And so after graduation, I ended up going to University of Michigan, where I did basic lab research in cancer chemical biology, specifically in pancreatic cancer. And what I found quickly, and I'd kind of seen this in undergrad as well, as I had an opportunity to do research, is that we in the lab were constantly discovering this novel cutting edge science. We were discovering new pathways, new molecules, new mechanisms, and yet those things often stayed in the lab. And because I was pre-med, I was also working as a medical scribe in the primary care setting. And so we were often seeing patients for which there were no medicines for the diseases they have. And so for pancreatic cancer specifically, as an example, since that's what I did my research on, gemcitabine was the current standard of care. It had been used since 1997. And before that, a different chemotherapeutic agent with a similar mechanism of action had been used, which was 5-fluorouracil. And both of those therapeutic agents work by inhibiting DNA repair and synthesis. And that's an approach that's been used since the 1940s. As you probably well know, there's lots of toxicity associated with this approach. And moreover, new therapeutic modalities have not really made it in pancreatic cancer. So the 5-year survival rates have really not progressed. And this was very sad to me. And it made me just notice that there is this two-sided problem. So on the one hand, there's this innovation gap where patients need new medicines. And on the other hand, there is this translation gap where innovations from the lab were not making it to the patients who could benefit from them. And so at that point, I became really interested in what could possibly serve as a bridge there for those two problems. So I ended up going to business school and getting my MBA from University of Chicago. I also had the opportunity to start a consulting business where I worked with multiple different biotech companies at the interface of science and business. I also spent 2 years with Arch Venture Partners, which is a venture capital group that started a lot of really well-known companies like Vir, Juno. They even started Illumina back in the day, which revolutionized DNA sequencing. And so really got to learn just the business methodologies that could be used to bring cutting edge science to the market where people could benefit from it. And just as one example, many of you guys probably have heard of Genentech, which is thought of in many circles as the first biotech company. It translated recombinant DNA technology out of the academic lab in order to create human-derived insulin in bacteria, which totally transforms diabetes treatment. As before that, it was made in animals, which, as you can imagine, caused a lot of side effects. So Genentech just serves as an example of how powerful biotech could be, because it managed to do that only 5 years after its founding, whereas we know traditional drug development can often take 20 years or more. And so I became very excited by this, and as Jane said at the beginning, became very interested in helping scientists understand what the best practices are in biotech to help make business approaches that will make biotech companies more likely to succeed be accessible. And so that's why I wrote the book. And what emerged as a central theme as I was interviewing multiple industry leaders was just this idea of building backwards. And it sounds kind of simple, so stick with me for a second. But building backwards is building with the end in mind, defining your end goal early on, and enabling you to more easily optimize your path to get to where you want to go. And this became something that stood out to me as I was speaking to people as kind of a pivotal mindset shift that you have to go through coming from an academic science background and moving into a biotech setting where there are these more defined end goals. And of course, you're probably thinking, building backwards, okay, that's all well and good, start with a goal, but science never behaves as we expect. Luckily, this is more of a method of thinking, so it's adaptable to any unforeseen scientific complication. And although it appears simple, it actually is very easy to botch, particularly in biotech. It can become especially challenging not to lose sight of larger end goals that are imperative to a business, as an entrepreneur is struggling as a scientist with the also challenging immediate goals common to a detail-oriented field like science. So we're going to talk about a couple different business strategies through this lens that can be used to help drive success. So I want to start with a case study of how this was done well. So there's this biotech company, it's called Synovion, and they decided to create a new schizophrenia medicine that was going to occupy a place in the market that prior drugs hadn't. And if we think about what makes a successful business, whether that business success is measured by successful clinical trials, ultimately being acquired, or having high revenue, any of those metrics often come down to creating a drug that is occupying a novel position in the market and doing something that other products have not previously done. And so that's what they wanted to do. And so they built backwards to what they wanted to ultimately do with their drug at the very beginning. And so if you know anything about schizophrenia, which I didn't when I first looked into this, it has two sets of symptoms, positive and negative symptoms. Positive symptoms are the abnormal perceptions, thoughts, hallucinations, and movement disorders commonly associated with schizophrenia. The negative symptoms are the loss of motivation or pleasure, dulled emotion, social withdrawal, and can often be equally devastating, if not more, than the positive symptoms. Existing drugs treat schizophrenia by blocking dopamine receptor D2 as the main target. And this has two problems. Number one, it tends to be less effective at alleviating negative symptoms. And two, it often causes a host of undesirable side effects because the D2 receptor is a GPCR, and those drugs often inhibit multiple different GPCRs, causing side effects. So Synovion sought to create a drug that would help alleviate both the positive and negative symptoms and also not bind D2. So how did they do that? You guys probably know a little something about drug development already, but just to review, typically the way it goes is you start with a novel compound that's binding a known target, and then you put it into in vivo studies to see if that effect plays out the way you think it will in a model. Instead, Synovion decided to reverse engineer using this kind of cool new AI technology where they were able to screen up front for a phenotypic effect on the mouse's behavior to see if it had an effect on negative symptoms. Then they could look at those compounds, and out of the compounds that seemed to work, they could then see if that compound was binding D2. And so through doing this compound called SEP363856 emerged, and it looked like it fit both of those criteria. And this compound acts on two dopamine and serotonin affecting receptors. So it also affects the dopamine pathway, but it did not bind D2. And by 2019, this compound had reached the notable milestone of entering phase 3 clinical trials, and results from the trials had demonstrated that treatment with this compound leads to a significant improvement in negative symptoms compared to the placebo. Additionally, the compound had lower rates of side effects as compared to currently approved drugs that target D2. So overall, the findings affirmed what the researchers had set forth as a goal for their new drug. So because this drug looked like it promised business success, pharma company Otsuka paid Synovion $1 billion just for the rights to co-develop this molecule. So although the drug has yet to complete its phase 3 trial and receive approval, it's on its way to differentiation in the market and has so far made significant strides to improve the quality of schizophrenia treatment. So I want to highlight that they were able to do this by this building backwards thinking through both, being able to think about where they were trying to go. They were able to carve out their market placement early on. And by using that method, it became easier to understand where novel technology could be useful. So really quickly, we're going to touch on a negative example of what happens when a company fails to build backwards. So a goal in biotech can be thought of as advancing through drug development in a way that progresses systematically towards drug approval. As a lot of biotech companies don't actually go all the way to approval, it's mostly important just to think about progressing through the process in a way that increases the value of the company as you go. So M Therapeutics was a company developing a new drug for stage 4 cancer. And they were able to go through this compassionate use pathway because they were using 2 repurposed, repositioned drugs. And they were able to show that they could bring terminal cancer patients into remission, which was really exciting. Whether or not this is a good foundation for the biggest biotech success ever financially is a different discussion. But regardless, the point still stands. So although the drug combination succeeded, the company ultimately failed because it ran out of money without substantially increasing the value of the business by initiating a clinical trial, even 6 years after its founding. So why? Why did that happen? M Therapeutics failed to build backwards. They were very focused on a step in the process, which was obtaining publication-worthy in vivo animal data, which of course is fundamental to the process. But as they had already been in humans, this was potentially a less effective point for them to spend time and energy, at least to the extent that they did, which was hiring 20 to 30 full-time people, resulting in a very high burn rate that was probably unsustainable. And they could have instead invested that capital into the necessary IND enabling experiments that would enable them to bring their drug candidate into the clinic. So again, M Therapeutics became so focused on a step in the process, obtaining preclinical data, that it lost sight completely of the broader end goal. And especially because they had this human data, which of course most companies don't have, they should have had an even more streamlined path through the animal data process. So those are just some examples of the power of building backwards and what happens when we lose sight of that in scientific companies. So one area that I think building backwards can be particularly helpful for scientists is when thinking through financing. So one way is by thinking through an exit. So you've probably heard this term before, exit in startups. And an exit is often thought of as being one of 2 things. So an exit could either be an IPO, which has a big asterisk next to it because it's not actually an exit, but we'll come back to that, and a merger and acquisition. An exit is important to think about early in a startup's life because it is thought of as a startup's path to liquidity. So it's the moment in which investors and entrepreneurs begin to realize the majority of their financial return. And if you don't build backwards to an exit from the beginning, that's often problematic in building companies because investors want to understand where you're going to go and where you're going to build value. And note that I'm focusing primarily on biotech startups. This is a little bit different if you're thinking about big, already publicly traded companies like Pfizer, for example. But in any case, a very generalized version of the process goes something like this. So a biotech company is founded. Investors invest cash in the company grows and develops, and then a path to exit, either IPO or M&A, and through that process, cash is returned to investors and entrepreneurs. Note that this is a bit oversimplified because sometimes you can exit without literally giving cash, like for example, you can get stock, but again, we'll touch on some of these details shortly. So we already talked about the 2 types of exits. So just to go into a little bit more detail, an acquisition is when a strategic buyer acquires the company, or that's the sort of exit we're talking about in the case of biotech. And to read this quote that I really like that sums up the power of an acquisition, this is by Bruce Booth, a partner at Atlas Ventures. A reality in biotech is that it's hard to escape the gravity of the balance sheets of larger firms. These big firms need external innovation to feed their pipelines and sales forces. The cycle of life often makes sense. The biotech takes the early stage R&D risk to bring new product candidates forward, and bigger firms, so like Pfizer, J&J, et cetera, take the later stage and commercial risks to get those patients around the globe. This underpins the price behavior of a lot of biotech valuations, as there's expectation great emerging companies will get acquired. So I want to pull a couple of things from that. First is that it's advantageous to both big pharma and small biotech to think about acquisition, because a small company by definition is under-resourced as compared to a large company. And so big companies like Pfizer and J&J often get a majority of their drugs by buying small biotech companies. And this is good for the biotech company because the founders get a payout and they're able to see their candidate drugs advance into the later stages of development, which are often too complicated and expensive for them to do on their own. The second option, of course, is going public. So going public is often thought of as an exit, even though the act of going public is not itself an exit. And so what do I mean by that? This is important to understand. And as a scientist going into business, this was never clearly explained to me. So hopefully this can be helpful for you guys. But when a company goes public, it's doing what's called a primary offering, which is when new shares are sold on the public exchanges. And those shares sold raises money for the company. And because you're issuing new shares, this also results in dilution to both the existing founders and the existing investors. So technically an IPO is closer to a financing event rather than an exit. But because your shares are now traded on a public exchange, this now gives you a path to obtain liquidity if you're an investor. So at that point, if an investor wants to get out, that's what's called a secondary offering. Founders and investors can slowly exit their positions by selling their personal shares on the stock exchanges. So now that is, giving cash back to them, not to the company. And a lot of times a big rule with that is that founders and investors have to hold on to their shares for at least 180 days following an IPO. And that's called a lockup period. And the reason that matters is because it's not like at 181 days afterwards, they're just going to sell everything and get out. First of all, that's a very bad signal. Analysts are often watching very closely who's selling their shares in the management. And if all of the management and investors sell their shares right away, that's going to freak people out and put a downward pressure on the stock price. So all that to say, for multiple different reasons, it actually takes hundreds of days oftentimes for investors to get out of a company after it's gone public. So it's not fully an exit, but it's a good path to an exit. So this is another quote by Bruce Booth that I like. IPOs aren't exits for VCs at all. They are just financing points on the long arc of building biotech companies. Given the limited daily trading of newly minted public companies, an IPO isn't real liquidity for any significant existing shareholder. It's a step along the path to progressively increasing liquidity over multiple years. So to go back to our thesis, building backwards. It's important to think about this early on for multiple reasons, because your investors are going to want to know where you're going and how you're going to get there. And as such, it's important to understand how an IPO exit compares to an M&A, so that you can be thinking about what exit makes sense for your company at every point in the company's life, because you never know when it might happen or make sense for you to go that route. So a couple different considerations. So one is the risk and return profiles. So often a lot of the value of a merger and acquisition transaction is actually in the downstream payments, so the milestone payments. And that is a good thing in some ways, because you're sharing the risk with your acquirer. They're doing, the phase 3 trial, or they're, taking the risk on how high revenue will be. But that also means you're sharing the reward. You're getting only a portion of whatever readout that is. In contrast, in an IPO, you own all of the reward, but you also own all of the risk. Additionally, time to liquidity is a consideration. So we just touched on that. An IPO can take a very long time to exit. And lastly, dilution. Going public can result in 20 to 40% dilution for existing shareholders, whereas an M&A doesn't have any dilution. So these are all important things to think about. So best practices is to really just keep an eye on which exit makes sense for you at every point in your company's life. And especially if you're raising money, have kind of a good answer in mind for every time you go there. But here's a good story I like about how this could look. So Vividian Therapeutics was planning to go public until June 2021, until they were given an acquisition offer by Bayer. This is sometimes called a stalking horse strategy when you're meeting with big strategic firms to try and get their buy-in into your IPO. And sometimes that results in an acquisition. So Bayer made them a big offer, $1.5 billion upfront for a preclinical company, which is nearly unheard of, and up to $500 million in milestone payments. And so one of the co-founders, Ben Cravat, said, and I'll paraphrase his quote, which is essentially the acquisition became a better path for them relative to the IPO, because with the IPO, they were still going to be having to develop all of their compounds themselves. And, there was kind of this focus problem of which compound could we develop. Whereas if they handed it off to a big pharma partner, the pharma partner had a lot more resources to develop a lot more things. So next, I want to talk about how valuation and understanding what this means can be important as a scientist. So valuation is a way of describing the worth of your company. And it's important to understand this for several reasons. So first is that you've projected exit value. So when you're meeting with investors, you can speak on that. Second, for the IPO process, you need to be able to demonstrate that companies similar to your own are able to achieve particular valuations. And third, it's actually a strategy to be aware of this early on, because something that I was told over and over again through my book interviews was just this idea of setting up a bidding war. So much like selling a house, you're going to get a better price for your company if there's a lot of people that want it. And so if you build a company from day one that has an asset and an indication that a lot of different big pharma companies like to invest in, that's good for you. With that said, there's an asterisk around you probably, if you're starting another anti-CGRP program, that might be kind of hard to sell given the fact that most large pharma companies are in anti-CGRPs already. But the point is thinking about which companies are interested in new modalities and which therapeutic areas they're in can be really helpful. So this is done in biotech through something called a precedent transaction analysis. Although there's multiple other ways to do valuation, a lot of them rely on revenue and cash flow. So I'm an MBA, I did lots of those methods, and they mostly don't make sense for preclinical biotech companies. And the reason is because preclinical biotech companies don't have revenue. You have negative revenue, very negative revenue. And so those methods just don't make very much sense. And they also rely on something called a discount rate, which is pretty arbitrary, as most drugs either really succeed and are blockbuster drugs or they don't make it clinically. And so a discount rate is hard to quantify. So the way you can think about this, and if you look at the image on the right, I did this for a migraine company, hence my reference to the anti-CGRP. And it was not an anti-CGRP, but you can see that there's multiple transactions and interests in neuroscience and in similar market indications. Additionally, it was an early stage asset. So I put the phase of development of each of those transactions in the phase column. And so you can kind of get a rough estimate for what different assets are worth at each stage of development. So next, that kind of raises the question of how do you want to think through, when building backwards, how do you want to think through financing as a company? And so I want to start by giving an example from a client company that I had at one point. So I had a client who wanted to raise $400 million very early in the company's life. It was a single asset company, which means they only had one molecule they were developing. And they wanted to raise for all 3 stages of clinical trials. And the founder, tells me he wants to raise 400 million. So there's a lot of problems with this plan, but let's just, hold on that for a second. So a conservative estimate is that this company has probably a $1 million pre-money valuation. This is kind of a conservative estimate for preclinical companies. And we'll talk about what pre-money means in a second. But what this boils down to is that a founder, if he does all of that he proposes here, he would be giving away 99.8% of his company. So let's think through what that means. So a pre-money valuation is just the value of the company before investment. You'll often hear business people and investors throw this terminology around. So if you're in the space, it's very important to understand what it means, but it's actually very simple math, especially if you survived physics and PChem and all of those various fun subjects. So to calculate the percent of your company you're selling with these metrics, you would think through the pre-money valuation. So like I said, that is just simply the value of the company before investment. So 1 million down here. And then you divide that by the dollars you're raising. So that would be the 400 million plus the existing value of the company. So 1 million. And then if you subtract that from 100, that will give you the percentage ownership of the company held by the founders. In this case, that boils down to 0.2% ownership. So like I said, not an awesome plan if you look at it through the lens of dilution. I will caveat this by saying that dilution is a normal part of growth, and the point of raising capital is because you're building more value with that money than you're receiving. However, with this quantity of capital in this instance, it's possible that they're raising more or creating more value than they're diluting their shares by, but it's probably not super likely. So without building backwards, dilution in your financing strategy can easily become too much. So how do we do that better? Building backwards to a financing plan needs to reflect the costs and timing of this process in addition to the realities of the investor marketplace. So something that's also important to understand is that the valuation, so remember that table where I showed you the valuation for a migraine asset, the valuation of a preclinical biotech company goes up stepwise in a function based on where a company is in phase of development. So normal companies, their value goes up gradually in line with revenue, but a biotech company actually goes up sharply based on where it is in development. And so you'll see here, and note the log scale, these inflections are actually a lot bigger than they look on the graph. The valuation of a company increases. And I put this together with my co-founder, Jacob Clanville, by analyzing recent data on the clinical phase of 154 biotechs that were both public and private. And so you'll see that there's this huge value inflection where a phase 3 company can easily be worth well north of 1 billion, whereas a phase 1 company can be worth well more than 100 million, which, is higher than the median for a preclinical company, which could be 100 million. Although note that the preclinical company are definitely skewed higher. Regardless, the point here is companies increase based on where you are in development, and that has implications for how to plan finances. So if you use a value inflection point, which is what I think of as those milestones to plan raises, if this company I had spoken with waited to raise money until after phase 1, where their valuation was perhaps 300 million, they could then give away way less of the company while raising the same amount of money. As I said previously, 400 million is still probably way too much for where they were, but pretending that's a valid number for a second, if we do the math again, now they own 42.9% of the company. And so my point here is if you are strategic about your timing of raising capital and think about where that will get you, you can actually build more value while holding on to more value as well for the current founders and investors. Note, of course, that there is a risk of underestimating your capital raises if you're timing it with the value inflection point. So you want to make sure you don't do that. But there's also a risk of overestimating, since that can, cause not only decreased returns, but it can also cause lack of focus and lack of discipline in a company. So examples of good value inflection points generally adhere to 3 different rules. Number 1, they increase the value of the company. Number 2, they decrease the risk of scientific failure. And 3, they demonstrate the future potential of the technology. So 2 examples of how this could look besides the obvious phase 1, phase 2, phase 3 readouts. Number 1, show the potential of a particular target or pathway to treat a disease. So this could be early data in vitro or in vivo models, highlighting the potential of a technology. The value there, of course, is solid data showing that this target or pathway might be relevant for a disease. So that increases the value of owning IP there. Second, it de-risks the company because it shows that the pathway's relevance in disease decreases the probability that the science fails down the road. Of course, science often fails, but it decreases the probability. And third, potential. It shows the potential value to building a company around an area. Another example of a value inflection point is a clean toxicology study. So toxicology studies are often where especially small molecule drugs fail. So of course, a successful toxicology study is valuable because it shows that you're one step closer towards the clinic. And that's an important component for an IND. So to start your clinical trials, you need that. Second, it decreases risk because it decreases the potential of down the road safety issues. Third, potential because it increases the value of a new platform. So for example, if you're going after a pathway and you have multiple assets that are targeting this pathway, if your toxicology studies show that it's not totally devastating to an animal to wipe out this pathway, that's very good for you and the value of your platform. So to summarize the presentation, building backwards is a key methodology in increasing the probability of success in biotech companies, both from a business and a scientific perspective. And it has the potential to augment your incredible science by providing it with sound business strategy that enables you to move more expediently and more at a higher probability of success towards the clinic. So building backwards to an exit and financing strategies are only two ways in which building backwards strategy can help de-risk a company early. So with that, I want to open it up for questions, and thank you so much for attending the presentation. And as Jane mentioned, I did just release a book, so I copied the cover art onto here because my friend did it and I think it looks nice. So, but I'd also love to connect with any of you. So my email is on the slide. So if anyone has questions, feel free.
Well, thanks, Stephanie. That's really great. I think what I really like about this is the idea that there's really a strategy built between the development, the science, and the business, and the inflection points. They change over time, and I think having those inflection points in mind really helps with the creative financing of a company, depending upon the stages and stuff. And sometimes that bar changes, and so it's always a bit of a moving target,
Right.
but I think it's always really helpful to keep in mind.
For sure.
So we'd like to open it up for questions or discussions. Feel free to open up your mics and ask questions of the group, or if you want to discuss a particular topic. Really, we're trying to focus this discussion around what are business strategies and how scientists and clinicians and business people can work together to build this company. I also think what you've talked about so far really demonstrates the kind of unity and strategic value between all three entities: clinical, research, and the investment community or financing. So I think that really demonstrates the kind of I guess unity between those groups in building a successful company.
Hey, Stephanie, my name is Consuelo. Thanks for your presentation. I was just wondering, over the course of your research, did you also look at the manufacturing impact in terms of building backwards and how that factors into the equation for valuation?
For valuation. Yeah, that's a great question. I did not specifically look into that question, but I can appreciate the impact of that. My company has gone through GMP a couple times, and our our P&L was hit pretty hard by that, so I can appreciate that. What I've generally found, and again, I don't necessarily have data to quantify this, but what I've generally found in speaking with investors is they kind of tie that GMP inflection point in with a phase one, where they kind of want to see, okay, that's great that you produce drug and it's stable and all of that, but what we really care about is like the phase one data. So like produce what you need to get through a phase one. So unfortunately, especially with biologics, the bulk of your phase one spending is actually in GMP oftentimes, not in the phase one, but oftentimes what I've found is generally VCs want to see both of those things together versus just one. But again, I can't quantify that.
Thank
Yeah,
you.
and hey, Consuelo, maybe you could, maybe people could introduce themselves and say where they're from, or just briefly where they work or what they
Oh,
do.
yeah, sure. So I'm at Seattle Genetics, and I work with antibody drug conjugates. And
cool.
manufacturing is a huge, piece for that technology. But there's so many new technologies that are coming out, and some of them, highlight themselves as, okay, our manufacturing cost is low, but like you said, you still have to have the data to support, proof of concept and effect, right, as you move through. But the manufacturing thing is always kind of intriguing. So in any case, thank you.
Yeah, so definitely can relate to that. I would love to have that one all figured out because we're dealing with that right now. My boss is actually very excited about building our own GMP facility to like decrease costs down the road. So we'll see how that goes. But
Yeah. Well, and I think GMP becomes critical if you remember what happened with Immunex. They were, had the fortunate problem of being incredibly successful and having a huge market. And Cindy, I think you were there at the time. And then they got behind in their manufacturing and unrealized some of the dollar value in Embryol because they couldn't manufacture enough as the market needed, which is an interesting problem. I guess it's a good problem, but it affected them somewhat negatively at one point.
Yeah, you never want to have a great product that you don't manufacture enough of. Yes.
Yeah, recipe for not success.
Yeah.
I
Yeah.
think it's also intriguing in terms of just building biotech companies and, the competitive landscape and figuring out like, okay, even if your drug is successful, if you don't do the competitive landscape homework, you might not be successful, right, Stephanie? Because that's part of that building backwards and including that competitive intelligence, which always is changing.
Right, for sure. And I definitely saw that when I was doing research for my book. I talked to this guy who worked at Abbott, and they developed a drug at Abbott for, I think it was hypercholesterolemia. I can't remember. But in any case, this was after Lipitor and stuff. So it was like a pretty crowded market. And they released the drug and it basically tanked. And then he did a bunch of market research and they looked at their clinical data, and luckily they were able to see that they met endpoints already that enabled them to change their labels. So they were actually able to change their label to include preventing high cholesterol in the first place, and that ended up being a huge success on the market. But had they not pivoted the label, the exact same drug was about to fail. So it's really, and they were very lucky, of course, that they didn't have to rerun clinical trials or anything like that. So it's just, I think, a lesson in what you said of how important it is to prevent that in the first place by building backwards to where you should put yourself in the market.
Thanks for that example.
Does anybody else have any questions or discussion topics that they want to add?
I was actually just kind of thinking of the various pitfalls, and maybe we can, discuss that. When you're in a young, small private company, you are a little bit more scientific or academia based. And some of the pitfalls is you've got so many molecules and you have to make a decision. And I have found that a lot of academic scientists, it's really hard to kill their baby. Okay. It's hard. It's hard to do no-go decisions at that point. And you have to have some good leadership there to help them along that path. And even in the clinical, because the fastest to the market is still has to be the end game. I guess that's where, you're looking backwards. You need to be on the market at a certain time. Otherwise, patent life, use patent life, everything. It is a race. And how to get people to understand this is a marathon, but it's still a race.
Right.
Yeah.
Absolutely.
and not to have people get in the various ways. I've been through the whole gamut now in a lifetime. There are so many classic pitfalls. How to get something out of preclinical that actually is safe. You can get it into the clinic, but it has to have a market. It has to be able to be a product.
Right. Right.
You don't want to comment on it? I mean, there's just so many pitfalls on where and how to help it along, so that in the end you've completed the marathon and you've won it.
Right. Well, I think that actually hits on something I mentioned earlier, which is just this idea that, you know, when I was working with as a consultant, I would oftentimes work with academic professors that were like, oh yeah, I found the next best XYZ. And, you know, especially early on, I do a lot of market research on their projects, or, you know, I talk to people in the marketplace, and a lot of times just the academia like landscape is very disconnected from the market, and they don't realize that. And so I think there's, you know, there's this movement now, and I think it's good to spin things out of the lab and to bring it to the market. But I think there's probably more of a reality check that needs to be there, to your point, than is often really there in academia, just because it is so separate. And so I've even found a disconnect between like university licensing offices in the market and that sort of thing, where they think, you know, something has the potential to be big, but I kind of know that most pharma companies view this field as like they've moved on from it. It was exciting 5 years ago, now they're over it. So to your point, it's constantly evolving, and especially academia is often just a totally different mindset, which is why I came up with building backwards as a succinct way to frame that, because a lot of times PIs are just excited about the concept and they're thinking very linearly about how, you know, big or whatever this could be, instead of reverse engineering. Okay, well, let's start with, does it actually have potential? And then what would it actually take to get there? Not what's the most exciting science necessarily. Like toxicology studies are not very exciting, neither stability studies, but you still need them to go to go into the clinic. And sometimes PIs want to spend too much time on the exciting kind of in vivo, in vitro work. Which is, you know, of course helpful, but you got to balance that.
So
sorry. Go ahead, Laura.
Yeah, so I'm Laura Ruthbach. I'm kind of different probably than most people here. I actually lead a disease foundation, and we're trying to develop, we fund research to try to develop potential treatments for a type of muscular dystrophy which has no treatment. And so it's interesting, this discussion is because we work a lot with academics. And so the hard thing is when they have a potential is how to get them over that hurdle, or to give them advice, you know, because I'm an academic, you know, I go, my training is academic, so I don't know a lot about business, you know, so I would love your advice about how you find the information you need. Like we have a researcher who's doing some preclinical work and has a good potential protein therapeutic, but how do we go about making it actually into a drug? You know, she can do all the nice academic work, but we're having trouble trying to determine what's the next step if we prove that it is potentially efficacious.
So there are a lot of think tank type organizations that take ideas out of universities or academia and do kind of that stage that you're talking to. And they kind of work with academics to help them understand what is the next critical pieces of information that they need to get then to draw in a biotech. And I completely agree with Stephanie. I've had the experience of working with some tech transfer groups at universities, and they want a huge amount of dollars for an idea that they really haven't validated. But this interim group can, you know, and usually it's small investors that are willing to put in some money, and then they usually have biotech business people helping them take these their think tanks that take academic ideas to a next stage that can get more funding, and then they would spin them out to the next stage in development.
Where can I find, like, you know, these
So the
kind
one
of,
in Seattle, which is escaping me, because we're in Seattle,
yeah.
and I'm sure everybody is above Grand Central Station I can't think of it. There should be, there's many of them.
I just wonder what was the names? I mean, I can, Google or look for things. I wasn't know what to call them,
Yeah, like for example, the University of Washington has one where they take ideas and they have a section within the University of Washington that moves it further. And Cindy, help me, what is the one in Seattle it's right next to, I'll look it up. I can send it to you, but, and then if you could talk to them or their CEO, they could probably point, where are you located?
Oh, I'm in Seattle. I'm right near North Gate.
Oh, okay. I can find the name of this organization. It's just escaping me right now.
Because that would be very helpful, not just for this one I mentioned, but in general, that's the hard thing we have. How do we translate what we're doing academically into potential treatment, or find companies are interested, or do other things to de-risk it so that companies would be interested? That's kind of the role of the foundation.
Yeah, and what these people do is they actually bring some money and they provide a lab space often, or they provide leadership to help that idea get to what you're talking about, which is the next stage.
Right.
So I will send that to you as soon as I find it online.
I would be curious in seeing what they're called in Seattle as well, since I'm not in Seattle.
Okay.
Be curious. But Laura, I would be happy also if you'd be interested to just, so we could go a little bit deeper, meet one-on-one sometime. My email's on the slide. But I'd say generally, yeah, an incubator or something like that is a good place to start. Also talking to various like VC groups and pharma companies, oftentimes even just talking to business development people, even if they're not going to hand you a check or write you a partnership deal, it gives you an idea of the market, which is what we've been talking about. So if you validated the science and you're talking to especially rare disease companies that have some experience in this indication, and they can kind of talk about what they're looking for in a data package, that can oftentimes be a bit more valuable than them writing you a check. So that's kind of what I've largely done, is just kind of start having those meetings and see what happens. So
Okay,
not necessarily, I mean, obviously getting money is good, but I mean, the foundation is willing to, pay for some of these early studies, to make it more attractive. We just need to know what's the best thing that we need to be supporting.
Yeah, so the one that I'm thinking of is called Accelerator Life Science Partners. And then the second one that I know of is called Commotion, but Commotion is specifically works with the University of Washington.
No, we don't fund anybody at the University of Washington.
Yeah, but look at Accelerator.
Accelerator Life Science Partners, is that what you said?
If we have a discussion offline too, and I can kind of look at specifically where you are, I'll be able to also give you more specific advice or connections as well.
Oh, I appreciate that. I've written down your email address.
So
I'll reach out to you.
I have somewhat maybe of a loaded question. And one of the things that's always perplexed me, because you pick companies that you want to work within, and some of those are based on the idea. Sometimes it's a combination of the idea, the funding, the leadership. Is it right for me? And so one of the things that I've always struggled with, and I've been in both spectrums, is who should be the leader of the organization? Should it be a business person? Should it be a scientist? Should it be a clinician? And my experience has been that individual changes over time based on the life cycle of the organization or the stage that they're at. So I wondered what people's experience has been, because an organization kind of needs different things at different times. And if you look at, again, Immunex, they moved away from a scientist more to a business person as they became more, business successful. And other companies have done the same. So I'm just curious what people think, because it really is critical to the success of the company to have this kind of collaboration between these entities, because it is a combination of business and science.
I would be super curious to hear people's opinions on this, actually.
Well, I may have a different of opinion, you know, because I come from a more of a patient advocate side. I've not been on boards and we've had not very much interaction with companies, but something that's difficult for us, we know the company has to succeed, right? And they have to do things that are good in business. But sometimes I wonder about if it's too much about the business and not enough about who you're trying to serve.
Yeah, that's
On the boards, because we were kind of an observer on a board for a company that was working, and it was difficult sometimes to be in the conversations because it was all about the business and how much money they could make, and not about the patients that they were trying to treat, you know. And I think there has to be a balance there, you know. And so if everybody's all about the business and you don't have anybody to advocate for the science or for the patients that you're treating, then I think that's a problem.
No, that's a great point.
That's actually something that I heard a lot, especially from people who did rare disease, that it was very fundamental to their organization and the success of their business to have that connection to patient mission. So I met with John Crowley. He's CEO of Amicus Therapeutics, and they do rare disease stuff. And previously, more personally, he had founded a biotech company to discover a medication for Pompe disease, which his two children had in the 90s, and there was no medication available for them at that point. So he discovered the first Pompe disease medication, or rather a scientist did, and he brought it to market through a biotech company from an academic, actually. But something he really highlighted to me was just how important it was to have that patient-centric mission, because it kind of kept them, you know, going back for new ideas and new therapies and wanting to get the best out there for patients, even though they do kind of have a monopoly of the Pompe market, so to speak. And so I think some businesses do that well, and probably some of them do it less well. So that's interesting that you bring that up as an observation.
Yeah, absolutely.
I think it's challenging. I think that CEOs really, they probably need to have a combination of both experience, because it's just becoming so complicated in terms of new therapies, the competitive landscape, the manufacturing nuances. To have a CEO that's purely financial without a real touch point for the science or the clinical development journey, I don't know. I don't think I could work for that company, because it's so complex. I mean, Jane and Cindy have been through so much, and to have a CEO that doesn't understand that journey would be really challenging for me personally.
Yeah, that's interesting. And you know, like I said, it changes over the life course of the organization. Is it Michelle? She has her hand up, I believe, or she's saying goodbye. I'm not sure which. Michelle, did you have something to say?
Or Michael.
I think it's
Hi, sorry. Hi, I'm a Southern California interloper here, but
No worries.
I wanted to join this group because I thought the topic was so so helpful, and I wanted to share, at least since nobody on this call knows me, but or nor may I be worth knowing, but at any rate, share with you that I've been in the industry for quite some time. I'm a former corporate vice president in the CRO world for 2 global CROs, as well as a VP of marketing for a couple different biotechs down here in San Diego. And I would really like to reinforce the need to be patient-centric about what we do today. And the fact of the matter is, Laura, in your efforts here, I offer to you also, you may want to reach out to some of the startup groups that are within Biocom or within California Life Sciences. There are groups in there of advisors to help, you know, the so-called 3 scientists in a garage. So you may have some opportunities to talk to some and network with some people who could get you to what you're looking for. But yeah, I just want to really reinforce, and Stephanie, your talk is really pertinent here, you know, kind of thinking with the end in mind and working your way back and understanding really what it does take to get to the finish line. It's so difficult these days because of not only the financial pressures, but there are a lot of societal pressures that are a part of bringing a new therapy, especially in the orphan space. I've been fortunate enough to be involved in bringing 4 different orphan drugs to market in the orphan cancer arena, and my involvement, 20-year involvement on the board of directors of 2 different orphan patient organizations, one in cutaneous T-cell lymphoma, really helped inform how I wanted to see both the commercial and the long-term clinical development pathway. And so all of you are speaking exactly to the right topic here. And thank you so much for letting me join and listen in.
Yeah, no worries.
Thank you for coming.
Yep.
Great comments.
Well, in the interest of time, I want to be respectful of people's time, and we moved it up to 4 o'clock. Hopefully did not encroach on people's evenings, but I know that it also probably encroached on people's workday. So I apologize for that. So if people want to send me or Cindy ideas, or do they prefer a later time or other topics, I'll put my email in the chat box. And I don't know if Cindy, you want to do the same, because we'd like to hear what you want to talk about, how we can improve this, other people that are able to join, etc. So thanks everybody for attending, and thanks to the EWIB, other folks of our Seattle chapter who joined, Lauren, and I believe Katie was on for a while, and Laura. Thanks for coming. Yeah, Cindy and I both put our emails in, so please email us with your ideas, improvements, because this is really supposed to help everybody network and get some value. And thank you, Stephanie, so much for your presentation. I know that was a lot of work. So
thank you for having me.
Yeah, thanks for joining us. I really appreciate it.
Anytime.
Awesome.
Let me know if I can join for the wine tasting next.
Yeah,
absolutely.
Awesome.
Thank you
Thank
very
you
much
so
all
much.
of
This
you
was
for
great.
coming.
Thank you, everyone.
Thank you.
Thank you.
Bye-bye.
Right.
Thank you. Have a good night.
Bye.
Everyone, bye.